lesson for this part and for today. As usual, I'm going to give a lesson and there will be an exercise at the end. But if you have questions or comments at any moment, please feel free to speak up and just interrupt me. And that's very nice to have these interactions. So what I want to talk to you now is about so you have seen how to determine protein structures and you have become aware that it is costly to do those. And so I'm going to talk about how can you predict protein structure. And to start with like a example to instill in your minds how it's possible to do what we are going to do, to use homology, for example, to compare structures. I want to start with a comparison between biology and geology, actually. So we have here something that is the tallest mountain on Earth, 8 kilometers tall, an incredible amount of rock, massive weight, and it has a very long age, and so it's like 60 million years. In comparison with biology, the geological scales look very stable and solid, and 60 million years ago there was no dinosaurs already. They had been extinct, but there were no primates yet, so life looked very different, and seems that things were moving on. And here we have this rock that was very, very stable, and I would say not that stable. I would claim that life has things that are much more stable. So even in comparison with this rock, life is more stable. This rock will be gone in another 60 million years. It will be erased, and it won't be there. And it was not there 60 million years ago. However, even in tiny bacteria, who lives maybe minutes, maybe a few hours, half a day, something like this, we have objects like glutamine synthetase, which is a tiny angstroms large molecule, which is doing an important enzymatic activity, so converting glutamate into glutamine. And this object has an age that is actually several orders of magnitude larger than the age of the Everest. So the geological scale is large, but life, it's more stable because we have objects that have larger lives than that, many orders of magnitude larger. How do I know that this object, this protein, has this age? How is this possible? So I know that this protein, it's very old because it's present in humans, for example, and this is an example of a PDB indicating us the structure of this enzyme. And I find this protein in many other species, actually probably in all species on Earth, and it's very similar. So this is the example from a bacteria, from Salmonella typhimurium, from another PDB file. And if we overlap these two structures, something that you are going to do later in an exercise, we can see that the core structure, the main features of this structure, these beta strands, these helices, are very conserved, and this is across a very large evolutionary distance. Of course, you have some differences, like in the loops, maybe this extra helix in the human sequence. Here you have some loops in the bacteria protein that you don't have in the human, because this protein, of course, is doing, although it's doing the same function in humans and in bacteria, obviously has to adapt to a different environment. Humans we are complex organism, multicellular, with different tissues, so it's not exactly the same, but the core structure has been maintained all of this time, And then this is indicating me that this protein has been around from as long as we have the generation of the three main lines of life: the archaea, the bacteria, and the eukarya. And since the Earth was solidified more or less around 4.6 billion years ago, the origin of life is estimated to have happened not really, very after. So 3.9 billion years, 3.5 billion years ago. And then about the last common ancestor, there are different estimations. Some will go to place it into 3.5, 3.8 billion years ago. Some that are a bit more extreme will go back to 4.29 billion years, which would be amazing considering the time the Earth cooled down. But in any case, even if we take the closest age, 3.5 billion years ago, this is indicating that this protein glutamine synthase and other proteins that are present in many, many organisms have been stably maintained in the tree of life, so that we have, and this is possible because we have had, of course, many duplications of this protein in the genetic material that has been passed from organism to organism. But also it's important to consider that there are some evolutionary constraints that have kept this protein relatively stable. And these constraints, they work at the level of sequence, of course, because you conserve the genomic material. There are mutations so that things can evolve and gain complexity. But also there are constraints because this sequence has to result into a structure which itself is constrained because it has to keep a function. In the case of glutamine synthase, an enzymatic function that is important for the organism. This protein has been, of course, multiplied and has evolved differently in different copies of the gene. And this happens in two ways. So we have speciation, which gives rise to orthologous sequences. So we have an ancestral sequence in an ancestral species. This, by speciation, you obtain two species. You obtain two species emerge from an ancestral one. Each of them carries one copy, initially very similar, of the protein, of the gene, which corresponds to a structure, which corresponds to a function. The function has to be maintained, and there's a relation. The structure has to be maintained mostly, but then the sequences they are going to keep some similarity. That is something that we can observe. The other way we have to generate new copies of a protein is by gene duplication, and this happens inside an organism. Similarly, we have an ancestral sequence, and then inside this organism, the sequence is copied so that, for example, we have the original enzyme, but then we have a second copy of the gene that could be discarded in evolution, or it can evolve to gain, for example, a new enzymatic function, which will be somehow related. The protein structure will be related to the original one. The general function will be related, but maybe there will be a difference in the catalytic center so that the enzymatic function could be different. But similarly to orthologs, we will have two sequences of two paralogs doing slightly different functions in the same organism, but the structures will be related, the functions, although a bit different, will be also related. And so all of these similarities can be studied, and you have seen a bit of this when we were looking at blast searches on Friday, by different bioinformatics methods that allow you to compare sequences and to represent these comparisons. So, for example, this would be the sequences of the glutamine synthetase for human and for the bacteria. Please note that the bacterial sequence is longer, so larger doesn't mean better also for proteins. And so in this case, in bacteria, although the bacteria is simpler, it has a longer protein. So it could be because it needs to interact with different things, for example, to sense proximity of nutrients or something, we don't know. So it can have a longer sequence because of the requirements, the particular requirements of Salmonella. And so here we would have a kind of a multiple sequence alignment, like when you do a blast search. And so you have the human protein and the bacterial protein, and like we were discussing the other day, the middle line indicating the similarities, and you can see that they are like, so we have gaps that have been inserted where the human protein would jump from here to here, and we would have like an extra sequence for the Salmonella protein, or we have the opposite here. We have this region with lots of conservation, many identical residues, and other, for example, this one here, this is a very long conserved region. And from these comparisons between just two species, we can learn that maybe this sequence is very important for the general function of the protein in any organism in the tree of life. But then we can learn about the specificities about what is bacteria specific or vertebrate specific or mammal specific and so on. And in case you have even, you can keep adding sequences, and we have many of those, you will see later in the databases. So for example, in this case, I would add humans. You have the human, then you have the archaea sequence, then you have the sequence from the yeast. And then with different tools that we are not going to discuss, there are web tools. I give you this one so that you can play with it if you want. You can make a multiple sequence alignment of these sequences. Gaps are added, each of the 4 sequences. But when you put them together, like in this case, and you will do this hopefully also in the exercise at the end of the lesson. And if you use some colorings, you can see, for example, that the insertion, so you can compare the 4 sequences, but it could be many more. Actually, it could be thousands of sequences. And so, for example, you can see that this region before that was very conserved between Salmonella and human, it's also quite conserved in the yeast and in the archaea. So we have this isoleucine, arginine, isoleucine, proline that are very conserved in the 4. So it looks like this region is important for more species. And also you can see that the insertion in Salmonella is really so far for these 4 sequences specific of Salmonella and is not present in the others. So the more sequences you add, and depending your choice of sequences, you can learn about the particularities of this protein in different taxonomical divisions. If there are any questions, like I said, at any time, please feel free to stop. So now, with structure, you have seen that that structure can be obtained by different methods: X-ray crystallography, nuclear magnetic resonance, and electron microscopy, which is gaining traction in the last years. What is important for us is that what I would like to note is that you have an increase in the PDB, in the number of structures by the years. So this would be, here is the time, and this is the number of proteins, of structures, sorry, that we have in the PDB. And the thing is that actually you would say, okay, we have a large number of structures in the PDB. This is very good. And it's something like 193,000, so almost 200,000. By looking at keywords in the UniProt database of sequences, I can find out that actually several of these PDB files actually will refer to the same protein sequence. So we have when I look at sequences, it's 56,000, something like this sequences. I would like to ask at this point, just to have a bit of a exchange. Let's see, how do you feel? Why there are more entries in the PDB? Why we have more structures than proteins for which we have some structure? Okay, so why there are some proteins in UniProt that have multiple PDB files? I'd love if someone would say something at this point.
Because there are many
Oh.
duplicates, many
Why?
duplicate sequences that code for the same protein.
This is one possibility, like as usual. Yes. So I will repeat this one. So you may have different splice variants from a gene, many sometimes coding for the protein, and then you could have PDBs of those. I hear two voices, so I would like the two of you that already said, I would like you to say something, even if it is the same thing. Please.
So maybe it's because of the interpretability of the radiology of the X-rays of the crystals, and thus different interpretations of unreadable areas, like we had in our very first protein structure lesson. Why we would have gaps in certain.
Not exactly. So if the, but I will follow up maybe. So if you have one PDB file, you have atomic coordinates, and then it's true, like you said, these atomic coordinates, and this is important. A program like Chimera will interpret those coordinates and make a representation for you. But the PDB will be one, and then maybe the different programs will interpret that data differently. So the coordinates are only one. There was a third person that I hear briefly. Please, a woman, actually.
Yes, I thought about maybe different isoforms of a single protein. So if we think about like the induced fit model, that we have one structure with the ligand and one without it, or
exactly.
Structures of multiple chains. So sometimes we have only one chain, sometimes we have multiple chains.
Exactly. So this is exactly, I just repeat. So you may have one protein, and then depending on what's bound to this protein, the structure may change. So then you may have different PDB files. The other one is the fragment, remember. So you may have different parts of the protein. So if you cannot solve the structure of the whole protein, you may cut your protein by domains or something, and then you may produce a PDB file, a structure of the N-terminal domain of the protein, or of the C-terminal 3 domains, or something like this. And in the end, moreover, anyway, some people may do the same structure, same quote, again. For example, imagine someone makes an X-ray with a resolution that is relatively good, but then after the years, someone says, oh, I have a better technology now, I can do better crystals. I'm going to try to do a new X-ray crystallography of exactly the same protein or fragment, same conditions, but with a better resolution. And so when you are looking for PDB files of a protein, maybe you can check the resolution and take the one that is better. Okay, thanks very much for the feedback. This is nice to have. But here we are. So we have the PDB is showing us structures for something like 56,000 proteins. However, in UniProt, if you query, so you know, give me the size of how many sequences you have. We have 231 million sequences. It's a lot, It's very useful because we can do all of these multiple sequence alignments and learn a lot. But it means that we have structural information of only 0.008% of all the proteins we know. And this is not too surprising. The point is that it is very simple to obtain sequences from organisms because sequencing has become extremely cheap. I mean, you can, sequence your own genome for hundreds of euros. It's something that is available to everybody, and of course, sequencing bacteria, archaea, small organisms. It's very, very, very simple, and we have all of these sequences of proteins that are derived from the genomes and maybe never were obtained experimentally. So we have only the sequences as derived from the genome because we obtain the open reading frames computationally. All of these sequences end up in the database, and it's a very good resource. But for structures, it's not so easy to obtain the structures. You have to really get the protein and have a bit of luck, and it's the cost and the time that you have to invest is very, very high. So what to do? What is the solution? And we have seen a bit already what the solution is. If you have proteins that are similar, they have a tendency to have similar structures. And so we can use this to infer the structures of proteins for which we actually don't have experimental information of the structure. So this is, it was called a bit, the idea, it was called structural genomics. And the idea is that if, so to make it more visual maybe and to reiterate. So let's say that these are protein sequences put like in a space, like if they would be like stars in the space. And so, and the distance would represent, for example, this would be proteins of the same family. This would be another family. This would be a protein that is very different from other proteins. So the closer the proteins in this space, the more similar they would be in sequence. And we know the sequences of all of these proteins because that's very, very cheap to know. And now say that someone solves the structure of one of these proteins in this cluster over here. And the idea of structural genomics and actually of homology modeling is that now, even if I don't know the structure of all of these proteins and at a certain distance of similarity, so if the protein is not related, it's very different, I cannot do anything. But with the ones that are very similar because they are from the same family, they will be all, for example, glutamine synthetase, and I solve the structure of human glutamine synthetase, then automatically, well, with different methods, more or less complicated, I could predict the structure of the proteins that are quite similar in sequence. And you can and you can go and now solve another structure here, and then you cover other part of this space. And you can imagine that if the intuitively, if the identity is very high, then the structures will be very easy to predict and will be very similar. And in the borderline, it will become hard. And so right now, we could say that more or less like half of the sequences in the database can be predicted. The structures can be predicted by homology modeling. And here is just a graph indicating that when the identity between the sequence to compare is very high, you can do very precise models. And as the identity goes down, let's say to less than 80% and lower, then the models are not so accurate, but still they will allow you to do different functions, okay, with those. And so maybe to focus a bit more, the use of homology modeling can help you, for example, if you are designing targets for a protein, and we will approach this in the exercise in a moment. So imagine you have this sequence you are working on, which is extremely similar to another sequence, but similar, but there are some mutations, of course. So there are many identities, but this glycine is a leucine and this lysine is a glycine in the other protein. But let's say that you don't know the structure of your protein for which you want to design a drug, but you have the structure of the similar protein. And then you know, because you know the structure, you know the catalytic center and the residues involving the specificity of the catalytic center have been discussed. And then you do your model by homology because they are so similar. But then at the same time, you can compare the in the same positions, residues that are equivalent in the alignment will end up in equivalent positions in the 3D. And so you can see, these residues that are different, they modify the specificity of the catalytic center, and then maybe you will be able to design a drug that will complement these residues better than these ones. So this is a bit the idea very quickly. Also, another thing that allows homology modeling is to fit. So from electron microscopy, sometimes you have structures that are like very low resolution, and so you cannot see the details of the structure inside. But if you can do models of the sequence that are supposed to go there, then in principle you can fit them like in a kind of a 3D Tetris in the structure that you have complete. And so in the end, you will end up having an extremely detailed model of a large complex. Thanks to electron microscopy, you could not see this because it had low resolution, but by fitting predicted structures, you can find a detailed structure of something that is very big. So this can be very helpful. And then a small reflection. Remember that we talk about domains that are units that have separate function, folding, and evolution. And also, if you are working with a protein that is very long and you want to predict the structure, it's also helpful to go domain by domain. So if you have domain predictions, it may happen that one particular domain, if it's that domain is similar to a sequence in the PDB, then it's very easy to predict the structure by homology modeling. But maybe for a domain there is nothing similar in PDB, then you have to resort to the structure prediction. And the situation may be different for different domains. So for some domains you may have information, for some will not, and then you will have to do predictions of the structure. And so very to finish this part, structure prediction has a history, and in the beginning it was thought that even in the absence of homologs, we could be able to. So if you cannot do protein homology modeling, okay, so you don't have you have your protein and you have nothing similar in the PDB with the structure. It was thought that it would be possible to predict protein structure ab initio without the use of any information from any other protein structure by, just by looking at the physical properties of the protein and then trying to simulate the folding. But this has not been working very well. But now we have a revolution in protein structure prediction, where this ab initio prediction is combined with homology modeling, and even if you have very low similarity to homologs, with something that is called correlated mutations. And I explain quickly. So what is this about? So imagine you have, this is your query sequence. Over here, it's much longer, of course, but this is like a fragment of a multiple sequence alignment. And then we find in the database everything that is similar to this protein. So if we would be talking about the glutamine synthetase, would be all of the copies of the glutamine synthetase in every organism that we have sequenced, which could be thousands. So this would be, and then we do this multiple sequence alignment. So this would be in yeast, this would be in E. coli, this would be in a fish, this would be the elephant. So all beings in the human tree of in the tree of life for which you have a sequence, you put them, you align them, and then this has because we have so many sequences, we can find positions which mutate in a correlated way. That's why this is called correlated mutations. So maybe one has a particular, when one has a particular charge, the other has a different type of charge and the size is larger. Or when this is an arginine, the other is an aspartic. Or when this is a tryptophan, which is a bit more hydrophobic, the other is a valine, which is hydrophobic. You don't know exactly how this correlate, but you can find the correlation because it's a mathematical thing. You just need to analyze the multiple sequence alignment. And the reason two positions separated in the alignment may be correlating is or could be because they are together in 3D when the protein folds. So you can imagine that if this amino acid mutates to something very big, in evolution, a mutation in this residue to make something small will be favored because now this residue is taking a lot of space, and so this residue has to make more space, or the converse. Or if this residue is gaining charge, positive charge, it would be nice if this residue would mutate to something with negative charge so that they will establish a bridge to stabilize the structure. So this is all. This you can get the alignment and then find correlations and then infer from them that the correlated positions will be doing a contact in 3 dimensions, okay, in the 3D. And this can be used actually to predict, and it's being used to predict the structures with relative success. So we have a combination now, which is you use the homology modeling. Okay, so if you have a structure that is of a sequence that is very similar to your query, you are going to use it. In any case, you look for the correlated sequence variation in the homologs. And then you do this kind of physical optimization of the structure, which, it will not be helpful to predict the structure without these two things. But together, it will help us to refine the final structure that we are predicting. So this is all about predicting the structure. And so, in fact, the program, maybe you heard of this, the program that is doing this with more success is called AlphaFold, which was developed by a company which is called DeepMind in London that was bought by Google, of course, some time ago. This guy is the brain behind, and this is a diagram, basically, just saying what I did say before, so that it's not, there is like a loop in this process, but then in the end, your query is a sequence. You use the multiple sequence alignment, but then in the end you predict a structure. And this is nice. They will tell you, they will give you colors. I show you an example in a moment, where you can see the parts that have been predicted with high confidence, and the parts where they just don't know really the structure. And actually, these parts where you cannot predict the structure, not always, but normally correspond to flexible regions, where in any case you could not know the structure anyway because they don't have a structure. They are kind of wobbling and moving around. Okay. So how to access these predictions? This is changing very much because they are recently AlphaFold predicted the structures of all the proteins, the millions of proteins that we have in UniProt. So they will, but they have to see how they make this data available. This will change probably. If you hear this video in some months, it will be different. But currently at this point, for example, for human proteins, you can go to UniProt. And then on the one side, of course, you have if there are PDB files, you still can look at those. Those are experiments, so it's useful to have them, and they have helped actually AlphaFold to do the nice job they do. But then for at least now for each human protein, you have a prediction of a structure which will look like this one. I just put the numbers on top. And so you see they predicted this part, which is like in the PDB, this part that is kind of a core, but there is also an extra domain over here. There is some helix here, and then you have this kind of low confidence loops, which is just probably disorder regions that are moving around a bit randomly. Okay, but nevertheless, probably with some function. So you can, this is just for you when you get the presentation, so that you see where you can look at these models. You can run AlphaFold in a Colab notebook. Well, this if you want to check this, it's not very fast. You can also, the source code is available, but you need a lot of computational power and this space. So it's not, it will not work in your laptop, I believe. So we are not running this right now. Maybe sometime in the future it will work on your iPhone or smartphone, but we are not there yet. So now, if there are no questions, I would like to introduce an exercise to finish. I just wait, but I'm going to explain an exercise that I did for you to see how you can compare structures in a way where you would like to check if the information that you have for a particular structure on a molecule that fits the protein can be transferred to another protein. The example is from the SARS-CoV-2, and again is like Tom was saying the first day. We didn't change. The only adaptation of this example has been to change now that is SARS-CoV-2. Before it was another SARS. So we had already an important example because we thought that this was an important issue, and sadly we were right. So, but here we go. So the idea is that so you are doing some kind of structure-based drug design, and so you have an original protein that for which some kind of drug, in this case, is going to be a peptide, has been designed, and you are interested on a SARS-CoV-2 protein, and you would like to use that drug on your protein, but you don't know, will I have to adapt it? Is it going to work? So you want to try, and you don't have the structure of. Sorry, no, you have the structure, but you want to know if the catalytic center is at least similar enough that you could just go ahead and try the drug, and you think that is going to work. Okay. And so what we are using is the original structure is a proteinase 3CL-Pro, which was the structure was solved, and it's not from the human SARS, it's from a porcine SARS. So it's a SARS that infect pigs. And so this is an important protein because it's a proteinase that processes the polyprotein of the virus. So you would like to block it so that the virus cannot be processed and cannot infect because it will have no proteins available. And this proteinase, the activity is it cuts after this residue. And so what is I'm going to be a bit fast over here. I'm sorry. So we have the structure of the SARS that infects, of the protease that from the SARS virus that infects pigs, in complex with a peptide that blocks it. And we have the 3D structure of the SARS-CoV-2 proteinase. Okay, but we wonder, is the catalytic center similar enough so that this drug will work? Also, will block the protein I'm interested on. Okay, and so it's important to know that there are 2 residues, which in the SARS from pig, they are in positions 41 and 144. It's a histidine and a cysteine, which are forming a catalytic dyad, which actually performs the proteinase activity, and which are the ones that are blocked when you put the drug. And so the idea would be to compare the structures of these two SARS to see if the catalytic center is the same. So at this point, what I will do is to let you, you have the script and you go to page 20 and 21. So from there you can start and follow the instructions, because you are going to use YAL-View to compare these structures. And so maybe in 10 minutes I will catch up with you and show you how this is done. But I think it would be nice if you follow the steps by yourself and try to answer the questions, and maybe we have later a bit of time to discuss on those. But better I don't say anything and I let you work on it. But at any point, if you have questions, me or the helpers will be glad to go over with you and see. When note please that in the, sorry that I keep talking, in page 23 there was some typos, so you have to change, like it says in the chat at the bottom of the page. It says, so this is page 23 in the script. So you have to change 1P9U colon B, that should be F. Okay, and 1P9U G, that should be H. It's in the chat notes anyway, so you can find that. But other than that, the script is okay. So please try, and in some 10 minutes or something, I'll get back to you to see to explain you, and we can discuss the result.